to use microphones or not?
No, when the air conditioning comes on.
Yeah, okay. Okay, then, ¿me escuchan bien?
Sí.
Okay. Bueno, a few words in Spanish and then I'll continue in English, okay? Bueno, bienvenidos todos, buenos días, muchas gracias por haber atendido la invitación. Bueno, tengo el agrado de presentarles al doctor Stanley Malloy y al doctor Kelly Hughes. Ambos los conozco desde hace rato. Con el doctor Kelly Hughes tiene rato que me conocí en los laboratorios de las montañas rocallosas. Al doctor Stanley Malloy lo conocí cuando yo todavía era estudiante de doctorado, en el 2000 y cachito. Entonces nos hemos mantenido en contacto y siempre hablamos de la posibilidad de hacer este curso, de hacer estas pláticas, y siempre se quedó en el imaginario. Afortunadamente esta vez ambos fueron muy amables en haber donado su tiempo y parte de los recursos para hacer estos seminarios. Y bueno, antes que nada muchas gracias y bueno, bienvenidos a todos. So, turning on in English. So, thank you very much, guys. We really appreciate the effort you're doing for coming. And welcome to the Escuela Nacional de Ciencias Biológicas. And the star is yours. Muchas gracias.
So, I told everyone before the lab people came in that we will try to talk slow so that the English is clear. But if you have any questions, if we use a word you don't understand, if we're talking too fast,
We need a microphone.
Can you hear me?
Yeah.
Just check.
I tend to talk very loud, so if you can't hear me, just let me know. I don't mumble like he does. So any questions about science, language, anything, stop, okay? We are going to use the board instead of slides. And the reason is because it slows us down. By us writing it, it gives you time to think about it. It also gives us time to think about it. And so we're not going to do rapid slideshows. We're going to talk to you about some important things. Okay? All right. And I'm so sorry that I have to talk in English because my Spanish is enough that I can get by on the street. But it's not the same. Kelly, a little statement about Kelly and me working together. We've known each other throughout our scientific careers and worked in labs together. We've both been fortunate to work with some of the best bacterial genetics people of the last generation. And we like to share the kinds of things that we've learned. We have been very fortunate to be able to teach courses on bacterial genetics to people around the world. And we really enjoy being able to share these things. All right? So here's the basic idea. Today, lunes, we are going to talk about some real basic core genetic concepts. Right? You can't understand the sophisticated concepts if you don't have a grasp of the core concepts. Tomorrow, we're going to be talking about transposons and using transposons to make chromosome rearrangements. Okay? So that's getting into sophisticated bacterial genetics. And on Wednesday, we'll talk more about reporter genes. The people in the lab are using transposons and reporter genes already. But we'll talk more about them and how they can be used on Wednesday. And how we can use them to understand genetic regulation. On Thursday, we're going to talk about mutations, bacterial fitness, and bacterial evolution. How thinking about a single cell can allow us to think about populations of cells. And then on Friday, we're going to talk about how both of us have used genetic tools for biotechnology applications. So you can see and think about it in a different way than the basic research in the laboratory, in a way where you're making something that can influence people's lives. Okay? A lot of stuff. All right. So what and why are we doing this? How many of you read The Salvation of Doug? Yeah? Some of you didn't. I love this story. Antonio, you want to read it in Spanish? Sure. Even if you've read it, you should hear it again. We'll talk about it.
Let me just find it. El rescate de Doc, William Sullivan. En una colina desde donde se veía una fábrica de automóviles, vivían Doc, un bioquímico y un genetista, cuyo nombre no diremos, ambos jubilados. Cada mañana, con un café y cada tarde, con una cerveza, discutían sobre diversos temas y puntos de vista filosóficos. Durante sus conversaciones matutinas, observaban a los empleados en una fábrica para empezar su jornada. Ambos vestían ropa de trabajo y cargaban una lonchera. Otros iban de traje y llevaban maletines. Cada tarde, mientras esperaban a que se asentara la espuma de sus cervezas, veían automóviles completamente ensamblados salir del otro lado de la fábrica. Tras dedicar toda su vida a la investigación, ambos desconocían por completo el funcionamiento y ensamblado de los automóviles. Decidieron que les gustaría aprender sobre su funcionamiento y, al tener diferentes formaciones científicas, cada uno adoptó un enfoque muy distinto. Doc consiguió inmediatamente 100 automóviles. Era un hombre rico, como es habitual en la mayoría de los bioquímicos. Y los trituró los 100 coches. Descubrió que los automóviles se componen de lo siguiente: 10% de vidrio, 25% de plástico, 60% de acero y 5% de otros materiales que no pude identificar fácilmente. Se sintió satisfecho de haber aprendido los tipos y proporciones de material que componían cada automóvil. Su siguiente tarea fue mezclar estas fracciones para ver si podía reproducir algún aspecto del funcionamiento del automóvil. Como pueden imaginar, esto resultó abrumador. Doc dedicó largas y duras horas entre su café de la mañana y la cerveza de la tarde sin lograrlo. El genetista, poco inclinado al trabajo duro, como puede ocurrir en la mayoría de ellos, optó por un enfoque menos extenuante y menos costoso. Un día, antes de su café matutino, bajó la colina, seleccionó a un trabajador al azar y lo amarró de las manos. Después del café, mientras el bioquímico se subía a la cremallera de su mono azul, se ajustaba las gafas de soldador y encendía el soplete para comenzar otra jornada de trabajo, el genetista se paseaba por la casa, se preparaba otra cafetera y hojeaba el último número de la revista Genética. Esa tarde, mientras los automóviles salían de la línea de montaje, Doc, empapado por el sudor del esfuerzo del día, tomó un sorbo de cerveza y en cuanto recuperó el aliento, empezó a hablar de sus avances. He estado centrando mis esfuerzos en un componente que encuentro constantemente en la fracción de plástico. Se ve así. Dibuja la forma de un volante en el borde de una servilleta. Ahora lo he estado mezclando con la fracción de vidrio para ver si tiene alguna actividad. Espero que con la mezcla adecuada pueda conseguir movimiento, aunque hasta ahora no he tenido éxito. Creo que con un soplete más grande e incluso con un lanzallamas obtendré mejores resultados. El genetista solo escuchaba a medias, pues su atención se centraba en los coches que salían de la cadena de montaje. Observó que les faltaban ventanillas delantera y trasera, pero no las laterales. En cuanto el bioquímico terminó de hablar, los genetistas son conversadores muy educados, exclamó: hoy he aprendido dos cosas. El trabajador al que le até las manos, esta mañana se encarga de instalar las ventanillas. Y la instalación de las ventanillas laterales es un proceso independiente de la instalación de las ventanillas delantera y trasera. Al día siguiente, el genetista ató las manos de otro trabajador. Esta tarde observó que los coches se fabricaban sin los dispositivos de plástico con los que trabajaba el bioquímico, los volantes. Además, observó que mientras los coches se dirigían al aparcamiento, ninguno daba la primera vuelta y empezaban a amontonarse en el césped. Esa noche, para consternación de Doug, el genetista concluyó que los volantes eran los responsables de que el coche girara, y además que había identificado al trabajador encargado de instalarlos. Envalentonado por sus éxitos, a la mañana siguiente, el genetista le ató las manos a un individuo de traje que llevaba un maletín en la mano y un puntero láser en la otra. Era el vicepresidente de la compañía. Esa noche, el genetista y Doug, aunque él no lo admitiera abiertamente, esperaban con ansias ver los efectos en los coches. Especularon que el efecto podría ser tan grande que impediría la producción de los coches por completo. Sin embargo, para su sorpresa, esta tarde los coches salieron de la línea de montaje sin ningún efecto perceptible. Los dos científicos conversaron hasta bien entrada la noche sobre las implicaciones de este resultado. El genetista, que siempre había sentido antipatía por los hombres de traje, concluyó que el vicepresidente se pasaba el día sentado tomando café, al igual que los genetistas. No tenía ningún papel en la producción de los automóviles. Doug, sin embargo, opinaba que había más de un vicepresidente, de modo que si uno no podía cumplir, otros podían asumir sus funciones. A la mañana siguiente, Doug observó cómo el genetista, en un intento de resolver el problema, se dirigía a la fábrica con una cuerda larga para atar las manos de todos los hombres de traje. Tras una breve vacilación, Doug, el bioquímico, dejó las gafas protectores, el soplete y bajó la colina a trompicones para unirse.
Gracias. So, the idea of that story is not to make fun of biochemists. The idea really is that by doing genetics, you can understand things that are happening in vivo. Things that are happening in that factory that you can't necessarily see, right? And the reality of the situation is that geneticists and biochemists do much better when we work together, right? So if you are a biochemist, do not be offended. But I like that story because it gives you an idea of thinking that there really are different ways of imagining things, right? So when you take that and you say that, so what is genetics, right? So if somebody was to say to you, what is genetics? DNA and genes and the relationship between them, okay? Mas?
The study of genes.
Okay, study of genes. Right. So those things are absolutely true. You can define genetics as the study of genes and inheritance, right? Not just the genes, but how it passes on information. But it's not simply the DNA and the genes. It's not simply, if we have the genomes of everything on Earth, there's still some real core questions, right? And the analogy here is thinking about plumbing, right? So in plumbing, we have a bunch of pipes. And you can understand all the pipes, but you don't understand plumbing. Right? Because plumbers need to know about the flow of fluids, where it changes pressure, where the weak points are, where you need to go in and fix things, right? Plumbing's not just pipes, just like genetics is not just DNA and genes. It's much more complicated in terms of understanding the relationships and interactions between them. And that, I think, is a really important point, and that idea is why Bill Sullivan wrote that little story. Bill Sullivan, by the way, is a very distinguished geneticist. So if we say, what is advanced bacterial genetics, right? In this case, we're talking about ways of building toolkits that allow us to answer really complicated questions. Right? And so what we're going to do here is we're going to take the basic concepts from the first day and build them into these different tools that allow us to probe what's actually happening inside the cells. Right? So So when you say, why do we need genetics? When we have all of these molecular tools. We can sequence genomes. We can build genes, right? We can do a lot of things. So why do we need genetics? Right? Well, the reason is because of this idea that it allows us to test what's really happening in that cell. Not in a general cell, but in that particular cell. Let's say that is a pathogen causing a disease. Let's say that is an organism in nature that's interacting with plants and has agricultural importance. What's actually happening in it is the thing that we really want to get at here. Is the gene product, is it necessary and sufficient? Right? Is the phenotype that we see, is that due to this gene, or is it because this gene, in ways we didn't understand, interacts with this gene over here? Right? That takes more than just being able to study that gene, because we wouldn't know about what else it was interacting with. And knowing this allows us to build bacteria that can do certain things that we would like them to do. Right? So fundamentally, that's why we really need genetics, even though we have such sophisticated tools. So here's what we need if we're going to do genetics. We need to have variation. Right? So we need to have different genotypes. Right? You could say DNA sequence differences. Right? The other thing is, we need to have observable traits or phenotype. Now, when I say observable, in some cases, it may be the colony made by that bacterium becoming wrinkly, right? Something we can easily see. It may be, for those of you in the lab, on an indicator plate, the color that you'll make, right? It may be a variety of things, but we have to have something that we can detect to be able to follow that particular gene. And then we have to have a way of combining these genes. Right? And this is gene exchange. We need all of these tools to do genetics. How many people in here work on bacteria? How about viruses? Okay. Right. So in certain bacteria, the two of us work on salmonella. We have very good tools for all these things. In some bacteria, we don't have very many tools, right? But if you know about the tools that work, it allows you to build new tools to work with different organisms. Okay? And so that's one of the key things here that we need to think about. So let's start then. This variation is fundamentally mutation. Mutations can be defined in a lot of ways, but let's just think about it in a very broad perspective. This is a gene, right? This is the sequence in the wild type. So this is the functional gene. We can, within that gene, get a point mutation, change of one particular base pair, right? But within that gene, we can also get macro changes. So, for example, you may get a deletion that removes part of the gene. We can get an insertion that sticks a piece of DNA into that sequence, right? And then we can get a variety of other things. Within that gene, if we have a certain sequence, let's just call this A, B, C, we can get a duplication within a gene where we've added that same sequence again. Right? This is a duplication. Or we can get an inversion in the gene. So this is fundamentally a micromutation, a point change. And these are all macro changes in that sequence. And those differences have a real profound impact on what can happen here, right? So, any one of these kinds of changes, any of them, can result in a null phenotype. That is, loss of function. Okay? How would a point mutation cause loss of function? Let's say we're changing an A to a G. How might that have a loss of function?
It might introduce a stop codon.
Okay, yeah, it could cause a stop codon. So premature termination. What else?
Change a codon in the active site.
Absolutely, an active site mutation. What else?
Recognition. Changing the recognition site.
Yeah, the recognition site I think is kind of like
Or the binding.
Yeah, this could be active in terms of binding or in terms of activity. A number of those things are kind of like that. A key function of that protein. Right?
Could be a missense mutation, like change amino acid for another amino acid, and could make a structural change.
So it could be a structural change that destabilizes the protein. So the protein unfolds, it denatures, right? So it could be a number of those things. So just because it's a point mutation, you don't necessarily know what the heck it's doing. Right? Now, a deletion, an insertion, any of these things here can also result in loss of function. Right? So, loss of function mutants are the most common kinds of mutations that you find. And they can be of a wide variety of different types. Right? So all of these can be this, but let's go to the missense mutations. So we've already said some missense mutations can be null. What else might be missense mutation? I had a very good friend who was a premier bacterial geneticist. Right? One of the best person in the world who understood some key important things. And you know, he always said, if I wanted a passive audience, I would have become a television announcer. I like to interact with the audience so that we're thinking together. Okay. So, what about if it destabilizes but not completely, right? So maybe it might be a conditional mutation. For example, it might denature at a high temperature but function at a low temperature. Or the opposite, it might function at a low temperature, cold sensitive, not at high temperature. Right? The other thing it might do is it might result in a gain of function. And that can happen in a number of ways, but for example, it might change the substrate specificity. This is really important because sometimes you would like an enzyme that did something that no enzyme does. And this is one way that you can find those kinds of changes. Right? So there's a lot of things that these point mutations can do. And I bring that up because we're going to be talking a lot about transposons and insertions. But transposons and insertions are not a whole story. These kinds of mutations are also really important, right? So spontaneous mutations and non-transposon-induced mutations. Now, let's step back for a second. We talked about mutations. There's a bunch of different ways you can get mutations in DNA. Those mutations have different properties. Different kinds of mutations may be useful for different things. Okay, so how common is a mutation? How frequently would we expect a mutation? So let's play a game. Let's take, this is the genome of a bacterium, right? And let's say it's a typical genome of Salmonella. So let's say this genome is 5 million base pairs. Right? That's roughly the size of the genome. And an average gene is about 1,000 base pairs. Right? So let's say we were looking, just doing a search for a mutation in a particular gene. Right? We would expect that finding a mutation in a particular gene would be that, right? Or basically 1 in 100. Thank you. A million's a million in Spanish or English. Yeah, this is 1,000. 1 in 1,000. Okay, but there's a problem here, and that is that a lot of kinds of mutations have absolutely no effect on a gene. Right? Within a protein, you can change lots of amino acids, and the protein doesn't care. Right? It's only a certain number of them. So let's just say that about 10% of the mutations have a phenotype. Right? So now we have 1 in 10 to the 4th. Correct me if that's right. Right? Okay, so, and that actually, I rounded this off because it should have done this 5, not 6. And this. This. Right? You're so picky. This is why I'm not a mathematician, I guess. All right, so let's just say now, okay, we're looking for these mutants, and we can look, we have the ability to see 1 in 250 colonies on a petri dish. Right? So on a petri dish, easily you can plate out, and if you have 250 colonies, they're separated enough that you can easily see each colony. Right? So let's say we're searching those 250 colonies for a particular kind of mutation. Right? How many plates? How many plates do we need? We would be looking through a lot of plates here. Right? So let's just say, if we were really lucky, we might find something. We might find 1 mutant in 250 plates, but it might be even more. Right? This is the concept of screening for mutants.
synthesis is not an exact science. And I do a lot of things called rule of thumb. Okay? Rule of thumb. And that number, 1 out of 200, is just a probability. Yes. But if you want a greater than 99% probability of getting it, you have to do the actual whatever probable times 10. So really, if you really want to get it, it's 10 times the probability. That's a good rule of thumb.
In theory, 200 plates per one, but statistically, you may not see it. So you're going to have to do 2,000 plates. That's a lot of plates. It's expensive. Media costs money. It's expensive. The labor costs money, right? And even at the end, you may not find your mutant, right? So this idea of simply having a way of looking for these kinds of spontaneous mutations is not a very effective way to go. Now, let's just say that instead of looking for spontaneous mutants, we mutagenize. We treat these cells with a mutagen before we go through the process, right? And if we heavily mutagenize them, right? Let's say we mutagenize them to the point that we increase this frequency 100-fold. Right? Now we're going to be able to find them a lot easier, but every mutant we find is going to have about 99 other mutations on the chromosome. That may make a difference in the ultimate genetics of that cell, right? So mutagenesis can be really good, but this can be a tremendous problem, right? I belabor this, and let me tell you why, because a lot of people, when they're thinking about genetics initially, they think, I will screen for this mutation. I would just take and look for mutants with that phenotype. And in many cases, they do that because it's really hard to think of another way of finding it, right? But the work you do for this is much harder than if you sit down and do hard thinking about how you can develop a genetic selection, right? And this relates to something that Kelly said earlier, which is one of the key things of genetics is you have to love to think. So if we do a selection, selection is a condition where only the mutant will grow. So if we have a good genetic selection, we can take and on a particular plate, instead of putting 250 cells, now let's say we put 10 to the 8th cells. Right? Now on one plate, we're going to find multiple mutants. Yeah? So the key here is designing the experiment such that only your mutant cells grow. Right? And there's a bunch of ways that you can do this that are really good. Now, once you've isolated mutants with particular characteristics, you may then manipulate them and use a screen to look for other kinds of changes. And you're already doing that in the laboratory portion of this course. Right? So screens can be really valuable. For example, if you're screening for LAC+, you can look for whether or not it forms a color on a plate. That's a screen. But you only do that in the lab after you've selected for tetracycline resistance or ampicillin resistance. So you have a selection followed by a screen instead of just screening from the get-go. Make sense? All right. How many of you have already thought about all these numbers? These are really basic core kinds of genetic concepts, but one of the things that happens nowadays, we are so caught up in teaching molecular biology that we don't think about some of these kinds of things, right? So the idea here is, before we get more sophisticated, let's make sure we understand some of these key concepts. You gonna make a comment, Professor Hughes?
I think it's going to close the comment.
Oh, is it? All right. We started late. We're going to have coffee early.
Can I make one
Yeah.
simple thing?
Of course you do.
I just want to, things that I think about numbers all the time. And I just want to, and maybe Stanley was going to say this and I'm just interrupting him. But if you do a positive selection, and it doesn't matter what it is, and let's say in one positive selection, you get this, and you do another positive selection. You see where I'm going? You see a few versus a lot. And just by looking at that plate, what you know, right? Genetics is all about inference. And what you infer from these two selections, at the target, the DNA target is very small, and it's much bigger. So numbers tell you target size, right?
Yeah, so I was going to say that. I told you we've worked together a lot, so we have the ability to interrupt each other, right? Sometimes this means that you have a bigger target, and sometimes it means you built a crappy selection. Right? Okay? So that's why when you look at those plates, you don't want to just come in the morning, oh, I got some colonies, and take all these colonies. You want to say, does this make sense? And it always makes more sense after you've had coffee. Let's take a coffee.
Pues como empezamos tarde, vamos al coffee break. Entonces acá fuera hay café y algunas galletas y algunas frutas. Empezamos.
still, with the air conditioner, still okay?
¿Me escuchan bien allá atrás? Es que con el aire acondicionado, sino para apagarlo. ¿Se escucha bien? Okay. What if we put like microphone on over there? Put it up here. Like,
If anybody complains,
hola, hola. I'm just gonna put it here. Sure.
Hello.
¿Se escucha mejor o no? Es igual. ¿Se escucha mejor así? Pero no quiero usar el micrófono.
Gracias.
All right. Here are the take-home points for what we've been talking about this morning. A selection is worth 1,000 screens. Okay? That means one plate instead of 1,000 plates to find a mutant that you're interested in. Right? You know, if you could increase your salary 1,000-fold, I'll bet you would do it, right? That's a big deal. Another thing here is you get what you select for, but you don't really know what you're selecting for until you get it. Right? All right, this is where I ask my colleague if he would just sing us a little bit of a song.
I know what he wants.
I want him to imitate Mick Jagger.
So you probably don't know, but before Mick Jagger was a famous rock and roll star, he was a famous geneticist. Right? And he gave us a very important lesson. You can't always get what you want. No, you can't always get what you want. But if you select sometime, selection's fine. You get what you need. Oh, yeah.
So that's point number 2, right? And part of that point is when you look at those plates where you're selecting, you need to look very carefully. Right? Don't just accept them, you have to think about it. And then the other thing here is that genetics does not end with a null mutant. Right? We talked about the different kinds of mutants, what they can benefit you to do. And so it's important in genetics that you get this. These are the 3 simple take-home points for the stuff we talked about earlier. Right? Now, one of the real issues in doing genetics is knowing what you're looking at. So let's just go back. So remember, all right, this is our hypothetical genome. And we find a mutation that we're interested in. Right? But this chromosome might have many other mutations. Right? It could have mutations here, 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 here. Now, you can sequence that chromosome. Oh, I have a mutation. I know it's here. I can just sequence the chromosome to see if there's any other changes. Do you think that would tell you if there are other important changes?
Not necessarily. Not necessarily.
Not necessarily. Why? Well, even when you do comparison, here's the thing. This might be a change in a sequence we don't understand. So you would think it's not important even if it is. Right? And sometimes it can be a change that we wouldn't think would have an effect. Right? So very often, in a base triplet that encodes an amino acid, the third position can change and encode the same amino acid. Well, in Kelly's lab, they found that often that third position can have a big impact. Even though in theory it encodes the same amino acid, it has a big impact on the function of the expression and the function of it. Right? So you see a third base position. How do you know? Right? A lot of things like that can happen. Your sequence won't tell you alone. When you look at genetic studies, about 50% are unreproducible. That means you do a lot of work, you publish a paper, other person gets your paper, reads it, says, oh, that's really interesting. They try that experiment and they can't do it. Right? One reason might be because you did something sloppy. That's always a bummer. But the other reason, very often, in genetics, very often, is because you are working with a bacterium that the chromosome is different than what they're working with. Right? Let's say, for example, you're really interested in Salmonella typhi, causes typhoid fever. And there's somebody who studied this Salmonella typhi from Thailand, but you can't get their typhi. So you get one from Africa, and you study it instead. You think that that's comparable? No, they both cause disease. They may be very different in ways you don't understand. Right? And so this is really important. What this means, whenever you isolate a mutant, you want to back cross it into the parent strain. That means the bacterium that you had before you ever isolated this mutant. Right? So to do a back cross like this means that you have to have a way of genetic transfer. Right? So how can you transfer genes between organisms? Conjugation? Good job. So electroporation. Now, if you have this gene on a chromosome and you want to move it between organisms, it can be really tricky. With conjugation, one of the problems is you tend to transfer a big piece of the chromosome. So you transfer large segments. You can design the experiment so it's a smaller segment, and still it's pretty big, right? With transduction, it depends on the phage that you use, but the phage we use in Salmonella, bacteriophage P22, it transfers about 1% of the Salmonella chromosome. Right? So a pretty small segment. So by transferring that small segment, it doesn't mean that there might be something very close to this mutant, but it means that you're eliminating all of these other things. All right? Now, again, to be able to do that back cross, you've got to have a phenotype that you can select. Phenotypes needed. Right? But this is a really important thing. The other way that sometimes that people use often to understand if a phenotype is strictly associated to a particular mutation is complementation. complementation can be tricky because sometimes phenotypes are affected by 2 or more mutations. I'm not going to go into the details of complementation, but when we talk about our paper today, I'll talk about examples of 2 or more mutations. The most common issue here is suppression or synthetic phenotypes. And again, I'll talk some more about that when we talk about it. Right? So you need a backcross. That's a really important thing. If you want to do this, it's very important for you to construct a clean strain. And the other thing that's important is that you share your mutant with other people who are doing this, so that they have a better ability to reproduce what you did. All right. Now, when you do this, get into this. Okay, so we talked about a few basic things right here. Now what we'd really like to do is we'd like to give you some examples of how the 2 of us have used these as your basic genetic tools to understand the function and regulation of a set of genes, right? So we're going to go through those examples, but again, it's really important if we get to something you don't understand that you stop us and ask questions, all right? And the first case here is going to be Kelly. He's going to talk to you about a regulatory system that he studied as a graduate student.
This is my turn. Actually, I'm going to talk to you about a biochemical pathway and something about regulation. But first, a little background. Hey. They don't like Stanley. Okay, so these gentlemen first came up with the idea of gene regulation. That genes could be turned on and turned off, and that was using lactose degradation. Stanley writes better than me. In E. coli. Okay. All right. So what did they discover? There is a gene called LAC-I, and there's a gene called, there's an operon, 3G. Okay. This is beta-galactosidase. So that cleaves glucose, lactose, to glucose plus lactose. And now we can row. Y is transport. Okay. And A converts lactose to allolactose, which is a better inducer. Okay. So one thing we know is we look at beta-galactosidase levels. Okay. Without lactose, 1. With lactose, 1,000. That's a big induction, right? So when you give E. coli lactose, they will now turn on the genes. Yes? How do they do that? They do that, so I hope this is a bit of a review. Maybe not. They do that because, let's see, this is a repressor. Okay? And that binds in the promoter region to shut this off. If you have some lactose, there's a little bit around, there's a low level, say 1, 0.1 right? 1 out of 1,000. There's always what they call a basal level. And that's just enough for this A to convert the lactose to allolactose, which then off. Okay. So they came with this idea of allostery. So you have a form plus lactose that binds DNA. And then when you have an inducer that interacts with the lactide, it changes the conformation. You have what's called an allosteric change. There it goes. Great work. Beautiful work. So this is the basic, and when it comes off, now you have RNA polymerase. RNA polymerase, you needed to turn it on, and that's composed of alpha 2, beta, beta prime, and sigma. This one's called sigma 70. There's also an omega subunit. We're not going to talk about that. So normally sigma 70 recognizes in a promoter region there's a -10 and a -35. Okay. Sigma 70 recognizes things. But in this promoter there's only a -10. Okay? But there's an upstream activating site which is recognized by something called CAP and cyclic AMP. Okay, so there's a protein, an activator protein that recognizes here. Sigma 70 will recognize here. And cyclic AMP, when there's glucose around, cyclic AMP is low. When there's no glucose, cyclic AMP is high. So glucose is always the preferred carbon source. And so if it's around, you don't want to express all those other carbon source genes. It's only when you don't have glucose, now you can sense that and turn on expression. So they figured all of this beautiful stuff out. Yeah? Okay. How did they do that? One of the things that really helped them a lot was positive selections. Positive selections. So they created some really beautiful positive selections. And I'm going to talk about those. I'll talk about two positive selections. They used chemical variations. So, lactose, which is glue-gal. There's something like this. There's a gal that's fused to something called TON, thio-ortho-nitrogalactoside. And this is toxic. Okay? So what if you have plates where they have TONPG, thio-ortho-nitrophenol, sorry. So that's taken up by LAC-Y. And they die. So what's going to happen if they put 10 to the 8 cells down and they maybe see colonies? What are these colonies? They're LAC-Y mutants. They are mutants that won't transport LAC-Y. Of course, you should do it not in the presence of glucose because that's going to be off. You should do it in the presence of something like acetate, a lousy carbon source. Right? Nice positive selection. Characterized transport. Well, that's another one. We'll do another one. There's another one called phenol-gal. Another compound that it works with. And that's broken down by LAC-Z, or beta-galactosidase, to phenol. plus gal, and they grow. But phenyl gal is not an inducer of the lac repressor. So you can put them on plates with phenyl gal, and they can't grow. But what happens? You get mutants. Yeah? They grow. How can they do that? A little louder. My ears are getting older.
And there is a constitutive like C.
Constitutive lac Z. So there's two ways of doing that. One is you can knock this in. Okay? So lac I minus, and then you're constitutively turning it on. What's another way?
The rate of reagents and
Somewhere in the promoter operator region, right? That maybe doesn't bind lac repressor or doesn't need cyclic AMP anymore, something like that. So promoter operator constituent. Okay. Now, this is a gene. This is a DNA site. Which are these mostly going to be? If you picked one, what do you think it's going to be? Lac I. So this is, again, like Stanley mentioned, so this is about 1,000 base pairs. This is going to be on the order of 10 to 20. Right? So we'd have to look through like 100 of these to get one of these, right? Okay, so now they got to figure out, they want to look at the promoter mutants, but they got this problem. Right? How do they get rid of that problem? Anybody want to try? How do we get rid of this problem of all those lac I mutants?
With another mutant, lac I. With another lac I mutant,
Close,
lac IS.
close,
No?
close. Not another lac I mutant,
Okay.
but two lac.
Okay.
So if they make a diploid and they have this F plasmid where they can carry lac I, and then a chromosome that has the lac I. Now, if your target here is 1,000, now your target is not 2,000 because they're independent. So if this is 10 to the minus 3 to get, now to get rid of both of them, it's 10 to the minus 6. So now, if I do this selection, lac I plus, like 2 copies, right? And I do phenyl gal plus, what's the plate going to look like compared to this plate?
yeah.
So you might see something like this, right? Okay. So numbers, again, numbers tell you the target size. Big target, small target. All right. So that's sort of a leeway. So this was the background that people thought about. Yeah, it's so great to have these positive selections using these artificial compounds to allow you to do selections. Okay. All right. So now I'm going to talk about what I did. I love this stuff. Oh, the lac code, gene regulation. So that's why they got the Nobel Prize. So they discovered that genes were actually turned on or turned off, not just always made and always expressed. All right. So one of my PhD advisors discovered an enzyme when he was a postdoc. And that enzyme in E. coli, that's a little light. Okay, something dark. The enzyme he discovered is called DNA ligase. He called it the DNA joining enzyme. Right? Okay. And DNA, it seals that last phosphodiester bond. So it puts in a DNA phosphodiester bond, connects them together. Sorry, handwriting's together. Okay. But to put that phosphodiester bond in, it takes out a phosphodiester bond from NAD. So NAD, you've heard of NAD, nicotinamide adenine dinucleotide. I don't know how you say it in Spanish.
Not
That? Oh, okay. In French, it's ADN. So it is nicotinamide, ribose, phosphate. So that's a nucleotide, right? Ribose phosphate. And then phosphate, ribose, adenine. So nicotinamide, adenine, dinucleotide. Okay? And ligase cleaves this. So it uses that phosphodiester bond here to put it in DNA. So that's what he discovered. All right. So when I joined the graduate school, University of Utah, I wanted to work with this geneticist. And so this was a guy's name is Toto Oliveira. And the lab I was joining, this lab that I was working with Stanley, his name is John Roth. He's a famous Salmonella geneticist. But Toto, such a fabulous guy, such a fun guy. Everybody loves Toto. Everybody wants to be with Toto. So John said, we need to start working with Toto. How are we going to work with Toto? And so Toto was interested in NAD turnover. So John decided, let's study NAD biosynthesis in salmonella. And that's when I joined. So, let's make some NAD. All right. Start with aspartate, neoaspartate, another compound called quilinic acid, and that's it, right? Oh, it's got it. Right. Okay. It's going to be a long pathway. Okay, so this is a pyridine with a nitrogen. Okay, and now we've got to make it a nucleotide. So the next step is to bring in ribose and phosphate. And so when you bring in ribose phosphate, it usually comes in the form of PRPP. And you break off the PP, and that goes to two Ps, and that provides energy to drive the reaction. And now we get to, and it also loses that CO2. So now we've got nicotinic acid. This is an acid. Nicotinic acid, mononucleotide. NA, okay. And now, from there, we add, from ATP, the adenine nucleotide. We get nicotinic acid. And AAE, and then the final step is to put on glutamine, the ammonia group, to get the ribose, phosphate, ribose, NAD. Okay, biochemistry. See, Sam is right, I was in biochemistry. All right. So John wanted to study this pathway. Turns out that there are other compounds, if they're present, E. coli would much rather use. It's expensive to make these, but if you have, let's say, nicotinic acid, it'll be converted to NaN, and so it needs pyrophosphate. So it can take this up from the outside. So outside. Okay, it can also take up nicotinamide. Nicotinamide, and that gets converted to nicotinic acid. Okay, so you have to remove the ammonia. So it can take these up exogenous. I mentioned that NAD is broken down by ligase. Okay, and you cut it in half to get nicotinamide mononucleotide, and then it goes this way to nicotinamide and back. So you have this cycle. So that's what was known. And John wanted to characterize the genes. So this is phosphorylated, right? It has the, doesn't get in very well. This has two phosphates. It's hard to get phosphates across the membrane. All right. So we can look for mutants that require these guys. So nicotinic acid or nicotinamide oxyfrose. Okay. So if we block this part of the pathway, we can feed them an exogenous and they can bypass this earlier steps. Yes? But again, yeah. And you can look at those. People did that the hard way. So they discovered genes. This was before me. Working hard, like Stanley said, going through hundreds of plates. We found genes are called NAD-B, NAD-A, NAD-C. Why it's not ABC, it's because they named the genes before they knew the order. Okay. And so if you're a NAD-A, sorry, minus, you're a pyridine oxidase. Okay? All right. So we wanted to try to get this gene. So that was my job. All right, I want to find this gene. Okay? That was my first project. It's an essential gene because I cannot take this up. Okay. Oh, dang. That makes life hard already, right? Jeez. All right. But I remembered Jacob node, and it turned out there are analogs. 6-amino nicotinamide, 6-amino nicotinic acid. Okay. And if you feed E. coli these compounds, they will not express de novo because they say, oh, we're not going to waste our energy. So they can sense that these are available. This is all. But if we feed them the analog, that analog goes to 6-amino, 6-amino NAD, and we get no growth. Right? So what's no growth? What's another word for no growth? Another two words.
No colonies.
That's no colonies. And for a geneticist, what does it mean? What do you have?
A house. Negative selection.
Negative selection? Positive selection. So if we have no growth, we can select for growth. Yes? Okay. All right, so that's positive selection. So again, using this idea of what Jacob know, if you get analogs, you can use analogs to provide positive selection. All right. Well, now we plate these on 6-amino. Oh, this green is not so good. Just yeah. All right. Plate with 6-amino nicotinamide. Okay. Get these. Right? Okay, what are those mutants? What are those mutants? So I'm going to give you some gene names for this. They call this pyridine nucleotide cyclin. This is ligase. They call this pink. I think it's D. This pink A. This is pink E. So for this cycle, they name these genes. What kind of mutants would I get if I look for 6-amino-nicotinamide resistance? Anybody want to try? So, 6-amino-nicotinamide gets taken up through this, right, 6-amino. 6-amino goes through these steps. Go to what gene can I knock out that won't take this up? Anybody see a gene that you could, gene or two? These guys, yes. So what happens if either this gene, X, or this gene is defective? So if this gene is not there, they cannot take up the 6-amino. If this gene is not there, they can take it up and convert it to the 6-amino-nic acid, but that stops there. Right? Nice. So you expect 2 classes, right? Classes. What gene is this? He's resistant to the nicotinamide, but he's still sensitive to the acid. That means he can still take up the acid and go in. So this must be okay. So this has to be pink A minus. And this guy is resistant to both. And he's P and CB. Okay, so we can play these games. All right. There's 2 other mutants you can get. You can get mutations in NADB, which is this very first step, and you get mutations in NADB, which I haven't mentioned, which is here. Now, so you can get rid of these because you have de novo, right? Doesn't matter if they're gone, you get resistance, but they're still making NAD. Right? So they're okay. This guy. That's weird. This guy. Well, that's weird. So, what did we learn? We learned something that whenever you have a biosynthetic pathway, this is the end product. And at the protein level, biosynthetic pathways are subject to end product inhibition. Okay? That means that if you're making enough NAD, if you got plenty of NAD, high, it will go back and actually allosterically interfere with this enzyme, so it shuts it off. Okay? We got plenty of NAD. Almost every biosynthetic pathway has this feedback inhibition, end product inhibition. Yeah? So these are mutants that don't bind NAD. They don't recognize this feedback inhibition. So even if the analog's there, they're always turning it on, right? So they're always on for what's called de novo. De novo biosynthesis. So even if there's some analog going in, there's plenty of good NAD, everybody's happy. What about these guys? I mentioned that's an essential gene because you cannot take two phosphates across the membrane. But these are just leaky. So they're slightly defective. So that means they can't ever make a high level of NAD. It's always low because this is defective. And so because it's defective and this is always low, there's no feedback inhibition. So again, they know lots of data. So if I do 6 amino selection, what do you think about a loss of function in pink A? Mini? Is it a big target or a small target? It's a whole gene. Whole gene. It's just the part of the protein that binds NAD. It's still active. It's a very small target. These are leaking. Again, they have to be active. Otherwise the cell dies. So again, it's a small target. So, how are we going to devise a genetic selection for the screen? What if we want to just find these mutants with this huge background? Oh, okay. Huh?
Again, doing the implementation.
So this is where, genetic, no, that's a good idea, actually, not helpful. But in a simple genetic selection, followed by a screen, to lower the playing field. How do we do that? So, let's see what kind of race. Right, so the idea is at 30 degrees, we look for 6 amino nicotinamide resistance, and at 42 degrees, we look for 6 amino nicotinamide sensitivity. Okay. What does that tell you? What kind of a mutant phenotype is that? This is conditional. Okay. So this is temperature sensitive. Okay. These are rare. So these guys, this is where it gets a little tricky. If he's resistant at 30 degrees, let's say in pink A or pink B, is the protein active or inactive? It's resistant. It's resistant to the analog. It's not using the analog. The wild type protein uses the analog and they don't grow. So it has to be inactive. Okay. So this is pink A or B minus at 30 degrees. And this is the wild type phenotype, right? They're sensitive to the analog. So this would be pink A or B plus. Okay, so this is a cold sensitive genotype with a heat sensitive. I don't know why Stanley maybe talked about that. Phenotype. But cold sensitive mutations in proteins are really rare, right? So it's easy to think of a protein having a slight change that causes it to start to unfold, and then you heat it up, right? And it unfolds. But the opposite, that's a little harder to get to. So these are rare. Okay? So now we can do this selection, and we actually get 6-amino, 30 degrees resistant, 42 degrees sensitive. Then we get mutants in pink A, cold sensitive, with a heat sensitive phenotype. Pink B, cold sensitive, with a heat sensitive phenotype. Mad B, that are feedback resistant, but that change causes a TS phenotype. So it falls apart and they don't grow at 42 degrees. Mad D, we get something that's 30 degrees plus, but 42 degrees minus. This is a real TS. This is a real TS. And if we look, we can distinguish those, right? This is going to be 6-amino nicotinamide, 30, 42 sensitive. 6-amino nicotinic acid, 30 sensitive, 42 sensitive, right? He's only going to respond to the 6-amino nicotinamide. Now to this guy is going to be, doesn't matter. Because he's after both steps. Okay? This guy, he's going to be dead at 42. What if you don't, if this guy's dead, what does that mean? That means you cannot make NAD. So now, he is a pyridine oxytrope. So on minimal, at 42, plus nicotinamide or nicotinic acid, they will grow. They don't have this, but they can use the exogenous. Right? This guy, he is an essential gene. So on minimal, at 42, nicotinamide, nicotinic acid, no growth. Okay? So by doing this simple little trick of looking for conditionals, we lower the playing field, right? So now, what could be a big target becomes a small target. And now everybody's a small target. And now we can find those super rare guys. Okay? Cool. Sorry, that's genetics. Right? Genetics, selection, followed by a screen, TS, and then growth phenotypes. And that allows us to find these things and characterize this pathway.
So let me tell you why I wanted Kelly to talk about this pathway. You noticed he talked about a lot of sophisticated selections of genes. There were no transposons there, right? There were no reporter genes. A lot of genetics can be done without them, right? But this, the way he described it, he said, how did you do all that stuff so quickly? Well, it wasn't so quickly. Every time you found something, it led to the idea of the next thing. So this was really meant to give you an idea of how basic genetics can really take you through identifying a lot of really important things for a key fundamental pathway in the cell. Right?
Next question. Watch out with the just don't fall.
Don't fall.
I'm not going to follow.
Okay. I have a question, Kelly. So you selected for
6-amino resistance.
yeah, no, and for the mutants. So did you add any mutant gene or just
Didn't
just
have
for
to.
the okay, wait for the
Yeah, so I had maybe 300 to 500 colonies per plate.
okay.
Okay.
That's fine.
Sometimes I had 1,000. I didn't work with those plates because it's too crowded. Yeah, variation. Gloria-Del-Buck fluctuation.
Okay, got it. Thanks.
All right. So I was going to tell you about another example of this. It's a little less convoluted, but we're getting a little later, and so as I'm going to tell you basically, I'm going to shorten the discussion of the discussion paper today, but I want to give you a problem to think about. You've been listening for a long time. Let's do a problem. All right? So, one of the pathways that was studied in the lab where Kelly worked as an undergraduate researcher and I was a graduate student was on fatty acid degradation. And it was in E. coli. Right? E. coli grows inside the gut. Fatty acids can be a very important carbon source for it in many conditions. This is true for a large variety of organisms. And fatty acid degradation occurs by this pathway called oxidative
Beta.
beta oxidation. Called beta oxidation. And basically it takes the long chain fatty acid and ultimately converts it to CO2. Right? It's a well-studied pathway. The enzymes in that pathway had been identified. There were a couple aspects of this that weren't understood. One of the things is it wasn't understood how this was regulated. Let me give you just some growth things that were really important in terms of these studies. So this is growth on LB, rich medium. Okay? So here we'll have OB. Here's time. Right? So if you take a bacteria and it's grown fine on rich medium, and you take it from this rich medium, and now we're going to put it on minimal medium plus oleate. So oleate is a C18 fatty acid. But longer chain fatty acids are the important fatty acids that are associated with membrane lipids most often, and so they would expect it to be most prevalent in terms of the natural environment of these bacteria. When you take them from here and you put them on here, and again this is a simple growth curve, OD versus time. What you see is that there's this lag phase, and then they start to grow. They grow till they saturate. Now if you take these guys from minimum oleate, and you go back here and you grow them on rich medium, and you take them from rich medium and you do this, you get the exact same growth curve. Yeah? But if you take them from this medium and you put them on minimal plus oleate again, what you find is they start growing right away. Right? What do you think that means? Yeah. So it suggests that you have to have induction and adaptation. You have to turn on the genes to degrade oleate before you can start degrading it. But once you turn them on, if you keep them in oleate, they stay on. If you take them out of oleate, they turn off. So it suggests that C18 fatty acid is important as a regulatory molecule for turning on this pathway. Right? So there's another fatty acid that people used as well. I'm going to restart this. And the other one is C10, or decanoate. C10 fatty acids are not so common in the lipids of membranes. They're shorter than the longer chain fatty acids that are present there. This is OD, this is going to be LB. No, I'll make this. I don't like me changing my curves. So if we take bacteria out of LB, now I'm going to take them out of here. So they're going from LB to decanoate here. What we see is there's this long leg, and then they start growing. Okay? So if we take them out of decanoate, now we say, okay, let's take them out of decanoate, let's take them back to LB. Grow them again in LB. Now we're going to re-put them in decanoate. We're looking at OD versus time. And what we see here is that they start right away. What do you think that means?
There was no reversal.
It suggests that to be able to grow on decanoate, right, you've acquired a mutation. In this case, that mutation is in the repressor in a gene called FADR. Okay? And if you have a mutation in FADR, so it can't turn these genes off, they stay on all the time, so they'll start going right away in C10. If we take those guys and plate them in C18, they also go right away. So by requiring growth in decanoate, we have a way of selecting for a mutant that disrupts the repressor, right? And if you want to know if it could just be adaptation, this is a real simple laboratory experiment that will tell you, is it adaptation or is it mutation? Right? So, a simple, important kind of thing to know before you spend a lot of time studying.
I call my bros.
Yeah, cool.
My bros. Here, this for you. So I joined this lab as an undergraduate. I was a chemistry major. And I learned that a bromo group, bromine, was about the same size as the methyl group. So, you know, the fatty acids are CH3. And I was able to find a compound that was 11 bromo, sorry. So C10, fatty acid, decanoate. And you can buy it that had a bromine group. And if you have a FADR mutant, they would be constitutive for the fatty acid degradation genes. So this is called 11 bromo decanoic acid. And if we put E. coli on these plates, they die. So that was kind of exciting. So because again, this whole Jacobian null thing, eating analogs, so the idea is somehow this is toxic. Right. So then I thought, oh, okay, let's try putting on 11, sorry, not an H, 11. My last name is Hughes, so I'm going to start writing my name. Sorry. And I gave it an alternative carbon source, acetate. Acetate's a product of beta oxidation. And guess what? What? Okay, so what's that? So these guys failed to, they couldn't break this down. So the toxic compound was actually bromo CH2COOH. So if you knock out any of the fatty acid degradation genes, you didn't break this down to the toxic compound, but you can still grow an acetate. That was fun.
So, we can tell some other stories, but let me just give you an example of when I started. So I started in this lab as a microbial physiologist. I was not a geneticist, and I was really interested in another pathway called the gloxylate shunt. Right? So the gloxylate shunt is a pathway that if you grow on a C2 carbon like acetate, and this acetate can get converted to acetyl-CoA and enter the TCA cycle. If you go all the way around the TCA cycle, you give out 2 CO2s. So fundamentally, you wouldn't be getting any carbon you used to melt cellular materials, right? So there's this pathway called the glycosylation, and in 2 steps bypasses the CO2 evolving steps of the TCA cycle. Very, very important pathway in plants, and also in some pathogens that rely on lipids in humans, for example, in TB. So I was interested in this pathway, and I had isolated some mutants in this pathway, and I wanted to find a regulator of this pathway, right? Somebody had published a paper that said, well, when you get the regulator of this pathway, that regulator has a certain biochemical pathway of product activity, and you can only detect that activity by purifying the enzyme and doing enzyme assays in vitro. Right? So we selected these things with a derivative of acetate, and I found a bunch of the mutants that survived that derivative of acetate. I purified protein from 200 cultures of bacteria. Paper argued that every time they isolated a mutant that's resistant and they purify it, it shows this altered enzyme activity. I purified 100 cultures with the enzyme, 100 independent mutants. I assayed 100 independent mutants, and none of them had that phenotype. It was a falsified paper. And I don't know if any of you have done much purifying protein, but we spent a lot of time in a cold room with columns. It was a heroic amount of work to get no mutants. That is a biochemist / microbial physiologist doing genetics. It's not a smart geneticist. Once I had done this, I had learned my lesson, and we went back and figured out ways to get those mutants. Just as an example. All right. Can we talk about the paper today? Did any of you read the paper we described? We had a discussion paper. Oh God, I'm so sorry. The paper I selected specifically because it indicates how certain kind of mutations, called synthetic mutations, can allow you to find important things that would be hard to find otherwise. Right? Let me give you some background on this that's really important. So at the end of a little bit ago, I said, that there are certain kinds of mutations that you only see the phenotype if you have both together. Right? One type of these mutations are what we call synthetic mutations. And synthetic mutations require 2 different mutations in different genes to exert their phenotype. Right? And when synthetic mutations, sometimes these 2 mutations can give you a synthetic lethal, in which case you have a situation that looks like this. This is growth. This is if we have, this is gene A, which is functional. Gene B is non-functional, and the cell grows. Likewise, if gene A is non-functional and gene B is functional, it grows. However, if both gene A and gene B are non-functional, you get no growth. No growth, they die. It's lethal. Now there's another kind of synthetic phenotype you can get where it doesn't define fully growth or no growth. It just defines a certain phenotype. Phenotype may not determine growth, right? And instead of calling those synthetic lethals, we just call it a synthetic phenotype. That is, when you have the two mutations together, it decreases one particular activity. Right? Okay. So the reason this is important is because there are certain kinds of mutations you might be interested in where there are these two different genes that can affect the process, right? And because of this target theory, getting mutations in those two genes at the same time is really hard. And likewise, finding an inhibitor that might inhibit those two genes is really hard. Okay? So the idea here is to say, how can you use this to improve the process of looking for new antibiotics? Okay? Now, antibiotic resistance is a huge problem. As we all know, it's a global problem that's getting worse and worse all the time, right? So let's just briefly talk about ways that you can get antibiotic resistance so we're all on the same page. One way you can get resistance is by modifying the target. A second way you can get resistance is by efflux. You export the antibiotic in the cell. A third way that you can get resistance is by gene amplification. So, if one copy of the target gene is inhibited by the antibiotic, but you produce 50 copies of that gene in the cell, that's enough to overcome antibiotic resistance. Okay? And by the way, this is actually pretty common. Right? And so another way you can block it is by blocking transport. So all of these approaches, chemists for a long time have been taking large pools of synthetic organic compounds that look like, for all intents and purposes, they may inhibit these processes, and testing them to see if they can be an effective way of killing cells, right? And so this is done very often. You might take a pool of something on the order of 20,000, 200,000 different synthetic molecules that from chemical structures look like they might inhibit that compound. And it ends up very often in the process of looking for the compounds that work as an antibiotic or an inhibitor of resistance. It ends up in the end that you get zero. A tremendous amount of work to screen for those guys. Right? This is done by large pharmaceutical companies all the time. Okay? And it's not very productive. It's very expensive, and it has not proven to be very productive. So the idea here is to take advantage of this synthetic property. And the concept is really pretty simple, because we have a lot of antibiotics that are combination antibiotics, right? So if you antibiotic augmentin, which I think is a very similar name in both English and Spanish. This is a combination of a beta-lactam antibiotic this is your cell. Gram-negative cell. The beta-lactams inhibit the cell wall if they get here. Right? And so they can kill the cell, but clavulanic acid inhibits the compounds that normally would provide beta-lactam resistance. So you put the inhibitor together with the antibiotic and it works really effectively. But you're targeting two different things. Okay? There's a whole bunch of antibiotics that we have that are like this. It's been a pretty productive way of looking at things, right? So now we come to the situation where we have methicillin-resistant Staphylococcus aureus. Methicillin-resistant Staph aureus are resistant typically to multiple antibiotics. They're very difficult to treat and result in a lot of death around the world. Okay? They affect the cell wall of gram-positive bacteria, Staphylococcus aureus. Normally, you get inhibition by beta-lactams like methicillin. But in this case, the methicillin doesn't kill them. Right? The activities that methicillin targets are tied to structures in the cell wall called teichoic acid. And it ends up, there's two pathways that fundamentally can generate the key structures of teichoic acid that are needed for this. Right? And pathways are shown in pretty good detail in the paper if you want to look at them. But if you just call them pathway 1 and pathway 2. Right? So in this case, if you disrupt pathway 1, pathway 2 still allows production of teichoic acid. And if you disrupt pathway 2, pathway 1 allows production. Right? And the probability of finding an antibiotic that simultaneously inhibits these two pathways, because they're so different, is really low. Right? So here the idea is that when you start this chemical analysis, you begin with a strain that's a mutant for one of the pathways. It can still make cell wall just fine. Right? So the mutation's only knocking out one pathway or the other pathway. And now, instead of looking for drugs that can do both at once, you look simply for a new drug that, in this strain where this is the only pathway doing this, can stop that activity. Right? So, obviously, if you're going through this many compounds, and you have to find something that's hitting both things, it might be really hard. But if you're looking for something that only hits one of those things, it's a lot easier, right? So it simplifies this pathway a lot. So in this case, where it's very common for people to find 0 inhibitors, now they found a number of inhibitors that inhibited one of these pathways. Okay? That's fundamentally the value of using these synthetic mutations to find an important new compound that was difficult to find by simple chemical screening. Okay. Now, there's two things you can do. This drug inhibits one pathway, but the other is functional. So now you can go back into a wild-type strain and look for a second compound that, when added with this first one, inhibits this pathway. So now you're going to make an antibiotic that's got two different chemical molecules. Okay. But you had a way of screening for it and isolating those guys because of this synthetic chemistry. Right? In this case, they really got lucky. You read this paper. Because it ends up that, in fact, when you got this one mutant, it sensitizes you to beta-lactam antibiotics. It doesn't give you resistance, but it sensitizes you to them. So what that means, if you can get better beta-lactam antibiotics, which are a dime a dozen, it's really easy to find that second inhibitor. Okay? So look, this is a really important thing. It affects all of our lives. The thought process of using this synthetic phenotype allows people to solve that problem in a way that they never could have before. That's the take-home point. Synthetic lethals are more than just a genetic play toy. They can really have important practical applications. Right? Anybody have any other comments about that paper? The paper itself is reasonably complicated. It goes a lot into the chemical nature of those pathways. If you've read it, I think it can be a fun read, but if you don't have enough chemical background, it might be a hard read. So that's why we didn't go into it in exquisite detail. Now, the paper for tomorrow, you're going to switch?
Yes.
Yeah. There's a switch.
There's a switch. So I was originally going to talk about because I work on building of bacterial flagellum. Had a paper there, which is pretty sophisticated. But I learned that there's an interest in Burkholderia in this facility, and so I thought I would present a paper where we just look for antibiotics, new antibiotics in Burkholderia using genetics. And so I think that would be a more appropriate paper for this audience. So we'll talk about that.
So just one question. So we will also have the T-pop paper plus that one, or just the one you mentioned,
We
right?
can. Is there a teapot paper?
Wasn't there?
I don't remember. Let's see what we got for tomorrow.
Yeah, that's,
Discussion paper for tomorrow is.
there's two papers.
Oh, we have teapot.
One is yours and one is Fabian's. So we're changing Fabian's.
We're going to change the second paper, the Q et al., to a Burkholderia paper. We'll still do teapot, right? Right? And we're going to look for volunteers to come up and. No, we'll save that. We'll practice first, maybe on Friday. Those of you that weren't in the lab, you don't know that teapot is an entertainment vehicle.
Okay, so I'm gonna email you the paper that Kelly's mentioning, because you guys don't have it, so I'll send it to you in a bit. And then, so just to be clear, so we are reading the one that it's Wozniak. So for that you guys can understand one of the experiments we're doing in the laboratory, and the other one is the one that Kelly just mentioned. Okay.
And if I could just add, it might seem funny to switch in the middle of things, but I think the key thing here is it shows the use of genetics in a different organism. And so hopefully that will provide broader applicability. Okay?
So you mentioned that they were lucky. Can you explain a little bit more about that? I mean, they were able to find one out of 230,000 compounds. So that's a lot of experiments to do.
It is.
So, and
But they found one.
yeah, that's only one, just one.
I know, but the key thing here is a lot of times they'll go through those screening processes and they won't find. So for example, it's easy to find chemical compounds that might bind to a certain cellular molecule that's important in vitro. Easy to find. But finding one that not only targets that molecule but can get through the cell membrane, that can accumulate to the right concentration both in the bloodstream and inside the cell, and do its job is really very hard, right? And so it's an example of this is the selection. This is a screen, the other is a selection. Okay? That's the key.
made this spring, they proved the 230,000 at the same time. How they made it, I didn't get it.
So basically, the way you do this is you take your 230,000, you test them, you pool them, and test the pools. And any pool that looks good, you divide it up and you retest that pool. And then you divide it up and retest that pool until you get down to a pool that has a few molecules. And then you test that, right? Most of these kinds of tests, to be able to do that in that pooled way, most of the time they're done in vitro until you get to the last step. And in vitro versus in vivo is a big, big difference.
Because what it was hard to understand to me is that if they make this pool, how they know what is the one that works.
They don't, so here's the thing. They'll take a pool with a bunch of molecules. I'll just start with this many. And this, something in this pool has activity. So now they break it down to this pool, this pool, this pool, this pool, right? And they find in this that that this pool, and this pool have some activity. So from that they say, okay, well, it's most likely this compound. Only this, I started with 6. I didn't start with thousands and thousands, right? And the amazing thing, so when you think about what you need to do to break down those pools, it takes a long time because you have to go through each of these steps breaking them down, right? And
So I'm just curious, so they have a particular compound represent how many times and how many pools?
the compounds.
Not having read the paper.
Somebody have an answer? Anybody have an answer?
To my understanding, some of them are variations of some of them.
That's
so
right.
let's say you can do the pools like that just with the same molecule and some changes in the R group. So that's where, and then I think that's a
Yeah,
little
it
bit
is.
of data.
But I don't think they told us exactly how many pools. They told us how many molecules they got in the end, but not how many pools they went through. On the other hand, they're starting with a much smaller population of molecules than a general random screen. Right? So they were able to find more derivatives with a smaller starting set than you typically find with a larger starting set. Right? And it's all because of the assay that they were doing.
So it seems the key is to have the smallest number of pools to give you the least amount of work to get to the answer. Anyway, that's...
I think the answer is to have the most effective in vivo assay in the end that you can do. But the in vivo assays, even though they may be really simple, if you're going to set up, 20,000 of these in vivo assays, right? That's the amount of work. It's probably not feasible.
But my point is not everyone can afford a $2 million robot.
Oh, but this was not meant for us. These are companies that are looking for new antibiotics, right? This is a different level. They have the robots. They've been running these robots, and the robots have been producing no new antibiotics. Okay? By combining that chemical analysis with a clever genetic assay at the end, they have now shown success. And by the way, this paper talked about the use of this synthetic lethal approach for antibiotics. It's also being used for cancer cells to find inhibitors of cancer spread. So it's proven really effectively. And it's specifically because you're breaking down those processes so that you can find the phenotype that you want. And that is all for today. We're happy to talk to anyone or answer any questions. And mañana in la mañana, we are going to be taking this to the next stage where we're going to be talking about transposons, right? So Kelly's going to talk to you guys about how to find pools of transposon insertions, and I'll talk to you about how to do chromosome rearrangements. Okay? Great.
So just a few comments about all these. So later today, well, the ones that we're having in the laboratory, we're meeting at 2. That's the idea, that's at 2. So to continue the 2 experiments we're running. For those that weren't able to be in the lab, so we're gonna, tomorrow Kelly's gonna explain, like we said, the tip-up experiment we're doing. Besides, they both were gracious enough to give me the strains in case you guys wanna try them, a bunch of strains to do the same experiment, but with your own model. So that's also one thing. And the other is, well, Stan was gracious enough to sense that the first paper was really hard. So don't believe that it's gonna be like this, so we need to discuss a little bit the papers and all that for tomorrow. And I don't know, any questions, comments? Yes.
It's about the paper. So we really can say that the compounds they found are really useful, because later in the paper they mentioned that they found 2 other variations of the protein that they affect. So is it not just like we're going to promote the selection of resistance?
So the idea here in the end is they're finding things that hit 2 targets. So the probability of getting resistance with those 2 wild type targets. Remember when Kelly was talking about you have a 10 to the -3 probability of getting 1 hit and a 10 to the -3 of the other. And because they're independent, it becomes 1 in a million. So the potential exists that by doing that dual target, it decreases the probability of resistance substantially.
Yeah, well, is it really useful to use it? Because in the future they're going to get selected just the ones that are resistant, so we are going to do another.
Well, so recent. So the point is that when you look at bacteria, anything that you look at, if you look hard enough, you will find resistance. Right? We're never going to get a miracle antibiotic that's going to just never develop resistance. There are people who argue that we are going to, and everyone has been wrong. Okay? And when you think about the numbers, there's just so many bacteria experimenting with so much genetic change that we're probably not going to get that. But if we get it, and it lasts for a period, it may save a lot of people's life. And during that period, if we're working on the next one, we have the hope of maybe staying out in front of some of the serious problems. And I think it's going to be a race, a continual race to stay in that situation. Oh, let me say one other thing, and that is, this is all, once they found good ideas, they patented it, and I can't tell you anything else about what they're doing with those molecules, because it's all secret.
Paulina, you have another question.
Sí, no solo rápido, si alguien no se registró, me dice por favor para apuntarlo, porque es importante para su
Any other question? How about the sound? Just, okay, Orlando, can you?
I see. No.
Can you use the microphone to answer them? If you want. Thank you.
Well, yeah. In the article, you say that lethal mutations you have to mutate 2 genes. And in the article, there was 2 pathways involved in this thing. But it could be possible that 3 pathways or 4 pathways, and it's how it works in the experiment.
Having two pathways, having two. And it's reasonably common in biology to have two pathways that are similar and may converge upon each other. It's much more rare to have 3 pathways that do something in common, right? And so I think that by starting with 2 pathways where there is this kind of situation, first of all, it's easier to detect the redundancy, right? And so it just makes it easier to work with. If you, let's say you had a situation where you had 5 pathways and you had to go in and knock out sets of those pathways, I would say it's probably not the best target to be shooting for, because it also means you probably have more ways that you can get resistance. If there are 5 ways you can do the same thing, why not a 6th way, right? So, and by the way, this is also one of the, this is why gene amplification is such a problem with many antibiotics, is because gene amplification is non-specific, right? Gene amplification can happen very readily, and so it can influence many different antibiotics.
What would be some limitations of adaptating this methodology to mechanistic systems? Because the article mentioned that the general approach should be applicable to eukaryotic cells.
So in eukaryotic cells, there are redundancies just like these redundancies here. And so in eukaryotic cells, it's a pretty similar kind of thing, but fundamentally, in many cases with eukaryotic cells, they couldn't do this before. CRISPR-Cas9 has allowed them to make more specific mutations in some of those cancer lines, so that now they can try this approach. There are no anti-cancer drugs yet on the market or on the path that I know of that have used this approach. The more different ways we have of looking for anti-cancer agents, the more likely we are to find them. I think that's the thing. Right? And it's complicated because, remember, eukaryotic cells are diploid, right? So you also have to deal with that diploid situation. And so in many cases you have to knock out both copies of the gene to be able to do this. Right? A tremendous amount of work. Cancer is a tremendous problem for people, so it's worth the work.
¿Más preguntas?
Well, in the paper they say that all the essay was in vitro. But how about doing this in silico to reduce all the cost?
Oh, it's silico?
With bioinformatic tools.
So, in many of these cases, they run in silico predictions before they start doing this. Right? So, they are doing a lot of in silico work in terms of doing the modeling. And it's not only the in silico bioinformatic approaches, but AI is really helping to speed up those processes. So, it's likely that in the near future, if that approach works well, we may be seeing more things come on like it. And I want to back up for just half a second. I focused on something that affects human health, right? But these kinds of things can really affect plants and agriculture as well, right? It can affect a number of things like that. So it's not strictly human health. In many ways, we can use these kinds of tools.
Alejandro, question.
I got a question about some of the methods that the article mentioned. One method is about that increasing the expression of a target decrease the susceptibility to a compound. My question is, that is something that the people of the article discover, or is something that is common to apply method to found the target of a compound?
That ties to the general information we know about antibiotics that's accumulated ever since we started studying antibiotics. They did not discover that. They're just looking for new molecules that will work anyway.
¿Más preguntas?
So I Hi. So I have a question about the protocol, and after they do the screening or before, they also do a transposon sequencing. So that transposon sequencing has to also always be done with the screening, or it can be independent. Just do the screening and then get the molecules.
There are people that do both, but transposon screening narrows down the targets that you're looking at, and so it can also streamline the process. Okay, but there are many drug companies that do this. They do not have genetic approaches that are tied to it. Right? I think so. Normally, papers that talk about synthetic lethal mutations are published in biology-related journals, microbiology-related journals. This was published in the American Chemical Society journal. And so I think that what this will do is make the chemists who are doing this type of stuff more aware of the possibilities to collaborate on those other more biological approaches.
¿Alguien más?
It's about part of the discussion because they said that they have a lot of, I don't know if it's the correct pronunciation, but they said that no sense compounds. But how do they know that there are no sense or that they don't have a specific mechanism on the pathway that they are studying? Or and also the after that they said that classified them because also they kill both strains. I thought that we were looking for that, but I don't know.
So the first part of the question was, how do they know this not narrow aspect of the pathway?
Yeah.
And I don't think in this paper they showed us any evidence that that's true. I mean, I think that's their interpretation. They have another paper that followed this one. I don't think that other paper showed that either. I think that is their interpretation of the result. And it might also be partly because they're chemists who think about things a little bit differently.
Thank you.
¿Alguien más? ¿No más preguntas? So just some other announcements. So one is that whoever wants to have the copy of this, the event and all that, I mean the guys in the copier, they have the file. So in case you want to just go and ask them, they already have it. The other one is remember not to leave trash in the auditorium. So when we leave, just be sure to take yours with you and drop it in the garbage can. And I'm missing another one. Bring a sweater in case you guys are cold for tomorrow, because I think we're gonna be using the AC once, like often, like suddenly we're gonna turn it on. Suddenly it's really hot, so we just put it on. And the last question is, I know they don't like using microphones, but it helps. Maybe like right now, I'm really happy you guys started making questions. So that's what this is about, to make questions. So I'm really happy you guys decided to do it again. So we're gonna use them. So then for the questions, okay? Okay. So I think that's it. Thank you very much, guys.
Mr.